Hello everyone. So I'm Rahul Vishwakarma, a graduate student at CSU Long Beach, who will be presenting the topic Smart Contract for DNA-based archival storage. So along with me, we have Hemant, who is a senior test principal engineer at Dell Technologies. So this work is a collaborative effort of Hemant and me. Let's begin with the agenda. That initially we will just introduce how DNA is a storage unit as compared to the digital information, uh, other storage media, for example, NAND and other storage media. Then we move out to the contribution and the solution part. That what exactly we made the contribution and a very high level overview of the solution which we'll be providing. We won't be going into deep dive of the solution, but a overall description of the solution may help reproducing the results which we have obtained. So if and then after we will just show the implementation, which is smart contract based deduplication, that along with the reason that why we are using uh, deduplication, which will be like of a smart contract for pushing the metadata to the blockchain. And for the conclusive remark, we will discuss few other storage media which can be used instead of DNA and possible challenges that when we store the data in DNA and then when we read it. So coming to DNA as a storage device, it's not kind of a new media which is being used as a storage device. So it's been quite old, like in 1988. So it was Harvard University which stored the image of a just an image into a DNA. So and then they just read it. So storage and retrieval is not quite new. So there's a kind of a there's a kind of a still comparison as compared to the storage unit, which is NAND and DNA. So over on the left hand side, you can see a NAND unit, a storage unit, and on the right hand side we have make for the comparison that DNA. So it is just like we are mapping the binary value 0 and 1 to a triplet codons like AGC or GAC, or it can be any of the valid triplet codon. So just like when we have a just one storage unit, and we can just create an array of storage, NAND flash storage. In a similar way, we can also have for DNA. So it will be like some sequence of nucleotide. So there's always a limit to which we can practically synthesize this DNA string. Is like 100 to 200 nucleotides. So the sequence will go to 100 to 200. After like 100 to 200, it's become difficult to just read them again. Now, there have been a lot of attention uh, for use of DNA as a storage device. So these are just a list of a few of the organizations which include the industry and the academia which have been working on DNA. So this is just for the reference and the FII purpose. So I have just listed them out over here. Now let's come with a motivating example that why exactly we need to reduce the storage unit information and use blockchain as a metadata for storing DNA as a storage device. So current challenges is cost. The cost is still the cost as compared to the previous time. It has went down, but still we want more cheaper the storage cost. Another is the security. For example, even if our data is stored, we can just someone can hack into it and can tamper it and can do anything what they want. And if we store DNA at a single place, it is a single point of failure. For example, how about if it's stolen? Although it's a known that DNA can survive for like 1,000 and 2,000 years, that's all fine. But what if it goes at the wrong hand? So according to this, I have tried to provide a solution and a possible approach. So the first problem statement regarding the cost, we will try to use similarity-based deduplication with a delta encoding, so which reduces the number of bits which will be need for representing the information. And for security, we know that if we store the data in a blockchain, it's immutable, so they can't change it. Now, another is single point of failure. So for that, we have a decentralized storage of metadata in the blockchain. So the last bullet point, we will be focusing more on the coming slides. Now, let's see like a high level overview of the contribution which we are providing. So compared to the existing blocks in a blockchain, we will be providing a customized data structure for that. And we will also provide a method by which we are going to store the data in each of the blockchain, and how a new block will be created. We will be suggesting logic for that. And finally, we will explain that how exactly we have done similarity-based deduplication, which reduces the number of information bit to be represented. And also, this will help us do a random read. Now, I think we already discussed this information in the previous slide, that why exactly we are storing the metadata on the blockchain. So, like decentralization, security, and data immutability. So these are the prime factors for consideration. And also, it will be like forever incremental, that we need not perform much changes. And for random access, random read, it will be much easier for us. Now for the proposed solution, this is a very simplified approach for the storage of uh, biological data on a blockchain based metadata. So over here we will be having locality similarity hashing, which will be transformed to key value store, and finally it will be sent to actual storage, which is a dedupe store. So and once we get the dedupe store, we will store each chunks over a DNA pool, so which is a physically we will synthesize in the lab. So, and this will be our actual digital information. Now over here, the prime focus of representing the slide is random access, that once we push the data to the blockchain based on similarity based deduplication, we will be able to access them in a random access. And finally, the metadata will be stored in blockchain, and actual data will be stored in DNA storage pool, which will be our actual destination. And the conversion of digital information to encode nucleotide, I will cover in the coming slides. So here I have just tried to summarize how exactly the read and the write will be done. So the text are just a summary of this slide. Now coming to implementation. So implementation, we have not done any wet lab experiments, that actual synthesis of DNA in the lab, but we have done kind of a theoretical study, that the advantage of doing it, and the feasibility study. So for example, let's take text, Dell Technologies. So the first step will be converting it to a binary of base 2. Sorry, this will be like base, yeah, this will be base 2. And finally, so this text, we have converted it to binary data, and this binary data will be converted to base 2. And with base 2, we will be having a mapping table which will be of the nucleotide sequence. each nucleotide unit like A, G, C, T, and over here A, C, G, T, and we will just map it 0, 1, 2. So this is done for the ease of mapping it. We can, although we can map it to binary to nucleotide, but the study has showed that when we map base 2 with the nucleotide, it creates like much less nucleotide information over here. And finally, this will be mapped to a DNA sequence, which will look like this. And our main objective is to store this information into DNA and metadata on blockchain. So for that, we will be so we have some code snippet which we did, and we encoded DNA data into the metadata as blocks. So over here you can see all the sequence, and then after the data was encoded to a BAM file. So BAM is a binary alignment map file. So for this genomic data, and over here we have, for the demo purpose, we have used multi-chain, this blockchain, and here we are just pushing this actual file, this BAM file, which is the genetic sequence, to a blockchain, and we are just naming it DNA over here. So here the stream was processed and data was inserted, and if we see this, a block is created. Now coming back to smart contract for deduplication, that there were just one design consideration. So the actual motive was to reduce the number of bits for representing the information. So here bits is analogous to like how we will be representing with the triplet codons. And over here we will be using locality sensitive hashing and key value store after performing LSH. So both of them are good for indexing. And finally, we can retrieve the value of the candidates using the hash values. Now, this is the proposed approach of locality sensitive hashing and key value store. So LSH and key value store. So initially it will be a BAM file, how we encoded the data in the, how we encoded the data. So it will be binary alignment map file, and we will just push it to, and we will perform locality sensitive hashing, and finally a key value pair will be created, and after doing some pre-processing we will compress it, and finally it will be stored to a dedupe storage, and this information will be uh, encoded as DNA in storage pool. And here it's like a very simplified logic of like how a new chunk in the blockchain will be added. So they will initially they will perform a kind of a search for just finding whether the data is deduplicated or not, and only if the data is unique, that will be pushed to a new chunk of the data in the blockchain. Now, writing the data is all good, but coming back to reading it. So when we try to read the data, we have to synthesize it in the lab, like a wet lab experiment. And historically, we have also, it's a known fact that when we synthesize DNA, we have to create a multiple strings in a PCR, that is polymerase chain reaction. So what happens during the process, that we will be having some kind of a deletion, insertion, or maybe like duplicate entries are formed. So here our main objective is that once we have designed the system, we also need a mechanism using which we can minimize the error of like getting error while synthesizing and retrieving the actual data. So for example, our actual encoded nucleotide which has been retrieved from the metadata is this particular sequence, and when we send it for synthesis, so it will not just synthesize just this one, it will just synthesize a multiple of them, like polymerase chain reaction, right? So now with that we need to retrieve now. So this can be like a problem statement, like given these strings, like multiple strings, we need to retrieve the actual information. So which is aligned DNA string, which will be this string. Now, if we, so there are many ways which we can solve it. The first is like using the information theory in the digital information theory, but that will work, but sometimes it might fail. The reason being that this data is quite different as compared to binary. So binary is like a 0 and 1, and over here, if we will be having triplet codons, so we need to customize our method or to figure it out like what method we can use. So actually, this work is a in progress. So we are still just trying to do a feasibility study that can we use like a customized Markov probability based error correction code based on Markov chain, and based on like we will do some kind of a transition probability matrix and see we can retrieve the data or not. Now, another thing is that apart from like insertion, deletion, repetition of the sequence, deletion is something which we are taking more coverage, kind of a priority. So we need to design some mechanism using which we can at least handle like 20% deletion. If 20% of data is deleted, then how we can recover the original data? 30% deletion. So according to that, we need to design some codex, so using which we can just retrieve it. So this is still like a ongoing work, in progress work for us. So we are always open for collaboration. So if you feel like the work is interesting and worth problem solving, we can definitely collaborate. Now, so overall, the key takeaway for this talk will be that there's a need for designing a mechanism. So not the compression, but deduplication based, which is good for genomic data that we, and it will finally reduce the overall price of cost of synthesis. So for that, we need to design a data structure which can be stored in a blocks in a blockchain. And finally, for reliability and security purpose and decentralization, we will be storing the metadata of the encoded information in a blockchain. And in process of doing that, we will also design a smart contract for deduplication. And finally, like we just covered a high level understanding of how exactly we can do the probabilistic error correction code for just reading it again. So this was the overall key takeaway from the conversation. We are we are now open for questions.